We are going to learn about quinolone antibiotics. So, first, you see the difference between quinoline. This is the quinoline nucleus. We studied this. Benzene fused with pyridine nucleus is quinoline. Benzene fused with quinoline nucleus. Sorry, benzene fused with pyridine is called as quinoline. Suppose if there is double bond or a keto group, then it is called as quinolones. It is called as quinolones. Which position? Second position. Second position. Now the second quinolone in the molecule, it undergo tautomerism. See, you can see it. One of the hydrogen can go on attached to the. Here you see there is hydrogen here. There is hydrogen. Now this hydrogen can shift onto oxygen. Can shift onto oxygen. Now here one of the pi bond will shift to. Sorry, we will. So now, please, sir, let me tell here clear. Sorry. Now here yeah, that's what we were telling. Hydrogen can shift. See here, this is the hydrogen which we were exposing. This will go out and form. Will attach to the oxygen. Attached to the oxygen, then one of the double bond will shift onto the between this nitrogen and this carbon. Now there is release of this hydrogen, that is extra. So it undergoes tautomerism. So you get 2-hydroxyquinoline, sorry, 2-hydroxyquinoline. In the same way, there is also 4 quinolones. That means a double bond keto group is present at the 4th position. Now, our drugs, quinolone antibiotics, have keto group at 4th position. It also undergoes tautomerism. It also undergoes tautomerism and form 4-hydroxyquinoline. 4-hydroxyquinoline. So let us see. This is some introduction. So these are the first complete synthetic drugs. Till now, whatever we have seen, they are from the natural source. Now these are completely broad spectrum and bactericidal drugs. Broad spectrum drugs. So let us see. So some of the important points here, what we see is you can learn. See here, fluoroquinolones remained as integral part in treating gram-positive as well as gram-negative bacterial infections. Both, that's what I was telling. It is bactericidal drug and also broad spectrum antibiotic activity. They are very good drugs for these reasons. Efficacy is good, spectrum of activity, and good oral bioavailability. Bioavailability is also there. We can take oral route of administration. There was many issues with penicillins, cephalosporins. We studied. You can refer them, all this neighboring group participation, acid problem. This is not available with these drugs. And also, you see, good oral absorption. Now, in the classification, you can see it, generation of drugs. As the generation increases, first generation to second generation, activity is also increasing, bioavailability increasing. As we discussed, bactericidal activity, then wide therapeutic index. safety is good. With therapeutic index means toxicity you will find even with a small dose. A small dose you can find the toxicity, but you can give these drugs large dose, no issue. Tolerability is also there, so safety is concerned. Few side effects are there. Of course, for any drug, we know that there is no drug without side effects. Then one more important thing is that we don't depend on the nature, they are synthetic ones. So synthetic traceability, you can find you can find them, you can synthesize how much you want, you can synthesize how much you want. So that is also one of the advantage. Then penetration, you can see it, very good tissue penetration is present. So it is able to interact with the intracellular parasites. One of the important thing is that these drugs can able to interact with the microorganisms who are living in our cell. In a cell, they may be in the outside the tissue, then we can kill. But these microorganisms, chlamydia, mycobacterium tuberculosis, brucilli, they are the Loginilla. These are, what happens is that they live inside our inside our cell. So that's why there are also advantage. Now all these are atypical organisms. What are atypical organisms? The ones which does not give color to staining techniques. Staining techniques are called as atypical microorganisms. All these are, you can't differentiate between gram-positive, gram-negative bacteria because these ones doesn't have any cell wall on them. That also very good penetration, and they can interact with intracellular parasites and kill them because these are bactericidal drugs. The serum half-life has been optimized. You can optimize with the dose. How much you want, you can optimize. So these are the some of the advantages of these drugs. So even in the penicillins and cephalosporins, even in macrolide antibiotics, we started telling the problems, then advantages from the problems, how we got better drugs we learned. But here itself, we are telling that they are very good drugs, very good drugs. So let us see in next slide. First, we will learn how these drugs have been discovered. Has been discovered. So first thing is that these drugs were as the name indicates, in the previous slide we told, these are drugs which are obtained completely from the synthesis. What they done is that we know chloroquine, which is anti-malarial drug. In the synthesis of chloroquine, when they were synthesizing chloroquine, 7-chloroquinolone. See, this is the byproduct or impurity present during the synthesis. This is the byproduct or impurity which was generated during the synthesis of chloroquine. So during the synthesis, what they do is that when they're isolating, usually we do column chromatography. Then you will find what happened. You will, when you do column chromatography, for example, this is column you construct. Now you place your reaction mixture on silica gel. So whatever the reaction, after reaction, you add silica gel into the reaction mixture and dry it with the help of rotavaporator. There is an apparatus called rotavaporator. Now you drop that in the silica, which is in column. Now, when you are extracting, you see, for example, if this is a TLC plate, we optimize this extraction isolation process with the help of TLC plate. Consider this is a baseline. Now baseline, you put your reaction mixture and starting material you place and you place the starting material. Now you run the TLC plate. When you run the TLC plate, you consider that your starting material spot is this. Now, if the starting material spot not present in the reaction mixture, that means you indicate that reaction has been completed. Reaction has been completed. Now, when taking this, so what happens is that you may get 2 or 3 more products and you isolate them. Isolate them. That isolated product was this one. One of the product was 7-chloroquinolone. They isolated and they kept aside. Then when they checked it, it was showing antimicrobial activity. It was showing antimicrobial activity. What is the basic ring present here? You see here the basic ring here, quinoline. Now here it is quinolone. Why? Because it is having a keto group at the 4th position. That's what I was telling. 4 quinolones are present in our, is the ring present in our drugs. So further modification, the 4 quinolone was there. So further modification they done. They here you can see when they done further modification, further research to get a better drug, we got nalidixic acid, which is a very good drug even till today. You see, it was discovered in 1962. Even till today, we are using nalidixic acid in the treatment of urinary tract infections. It is still used for the last 30 years. We can tell 50 years also. It is still used. And maybe streptin they may use, but still we can use nalidic acid for the urinary tract infections. Now, when you observe the structure of this, now you can see it. It has 2 nitrogen rings attached, 2 pyridine nucleus presented, 2 pyridine nucleus. So this you can call it as what is that? It is called as, see, the basic ring is naphthalene. Yes or no, please? Naphthalene. You can also call it as diazo. 2 nitrogens are present. 2 nitrogens are present. So diazonaphthalene. You can call it as diazonaphthalene. Diazonaphthalene. Or you can also call it another name. Another name is 1,8. If you give start numbering 1, 2, 3, 4, 5, 6, 7, 8. At 1 and 8 position, it is called as naphthoridone. What is the another name? Naphthoridone nucleus. This is the nucleus present in nalidixic acid. Nalidixic acid, further modification we got our drug flumequine. What is here? You see, there is a fluorine group attached at the what is that, 4, 5, 6th position. This is the first fluoroquinolone antibiotic which they have discovered. Fluoroquinolones are more famous than naphthoridine derivatives. So, fluorine attached because it has changed its physical properties, absorption properties. That's why we also call this classification fluoroquinolones. But it has been withdrawn from the market because of teratogenicity. Further modifications they have done. So where they placed piperazine nucleus, which is ciprofloxacin. This is the now most successful fluoroquinolone. So further from the ciprofloxacin, we got various new fluoroquinolones. So it was that's why I told you in the first, it is the first antibiotic which was discovered completely from the synthesis. Completely from the synthesis. It was accidentally discovered. Which one? 7-chloroquinolone was an impurity. It is an impurity or a byproduct. Byproduct which was isolated with the help of column chromatography, and that byproduct they have checked for activity. They have checked for activity. Then one by one we got naphthoridine nucleus. Naphthoridine means this is naphthoridine nucleus, which one of them is nalidixic acid. Then they again went back to quinolone derivatives. Naphthoridine is only one drug. One more drug is there, at the later we can see it. Naphthoridine. Another, again they went back to quinolone derivatives. Then what is the first fluoroquinolones? Is flumequin. Flumequin, which is the first fluoroquinolone antibiotic. It has been discontinued because of teratogenicity. Then from this we got what is that? Pyrazine nucleus has been attached, which is ciprofloxacin, widely used. So this is how we got these drugs through the synthesis. Let us learn next classification of drugs. Classification of drugs. Any doubt, please? Any clarification? Hello?
Hello.
Any doubt?
No doubt, sir.
Okay. Are you listening or you're watching something? Please try to concentrate. I know you'll be distracted, but try, please. These are various generations. Drugs. The drugs are classified based upon generation of drugs. Let us see. While we are learning, we will see it. The first generation drugs are called nalidixic acid and inoxacin. Both of them have naphthoridine nucleus. That is diazonaphthalene structure. These drugs mainly act on gram-negative bacteria. Gram-negative bacteria, particularly in the treatment of urinary tract infections, and they are not able to distribute other parts of the body. So they were not able to kill the infections in other part of the body. They are concentrated in this in the kidney. So they were able to show. Both of them is having naphthoridine nucleus. Remaining second, third, fourth generations are called fluoroquinolones because fluorine atom is present on the quinolone nucleus. Fluorine atom is present on the nucleus. So you can see it. Norfloxacin, inoxacin, again, here some of them we kept here. Some textbooks they tell us first generation, some are telling us second generation. So don't get confused. Here that's why I kept both place. So again, ciprofloxacin, ofloxacin. These are second generation drugs. You can see it. They have enhanced activity 1,000 times against both gram-negative as well as gram-positive bacteria. So what happens as we are moving to generations? These are gram-negative infections. First, nalidixic acid. These are gram-negative bacteria. As moving to generation, they also show activity towards gram-positive bacteria and other microbacteria also. We can see it. So 1,000 times activity increased. 1,000 times activity increased. And effect on atypical organisms, which we told, that is that Mycobacterium tuberculosis, chlamydia, those microorganisms also they affected. Among all this, ciprofloxacin, a second generation fluoroquinolone, is most successful, both economically as well as clinically. So the one who discovered may have got lot of money. Then third generation, fourth generations, levofloxacin, gatifloxacin, sparofloxacin, fourth generation, moxifloxacin, gemifloxacin, and trovofloxacin. Many are there, few we kept here. So both of them, newer fluoroquinolones, all these are new, such as gatifloxacin, levofloxacin, all these offer enhanced activity against aerobic gram-negative bacilli and improved gram-positive activity over ciprofloxacin, even to streptococci pneumonia, prostatitis. In all this, it's an advantage with the third generation and fourth generation drugs. So this is classification of drugs. You can classify if you want naphthoridine structure and quinolone drugs. Naphthoridine structure, if you remove this, these are the naphthoridine nucleus. Remaining are quinolone antibiotics. What quinolone? 4 quinolone antibiotics. 4 quinolone antibiotics. Because keto group is present at the 4th position. You can also classify like that. Even the streptomycin drugs, aminoglycoside, we also classified like the streptidine and streptomycin, 2-deoxystreptomycin. Right? In the same way, you can also classify here according to structure. Next one. See, this is the nalidixic acid. What is the disadvantage? Let us see one by one here. We are almost done. See, we learned this. What is that? 7-chloroquinolone was the intermediate during the synthesis of chloroquine. So when they done it, 7-chloroquine during the biological screening found that it has limited usefulness due to high protein binding. This is having high protein binding. Clear? And approximately, see, half-life is 90%. Then, so it was so 7-chloroquinolone acted as a now lead molecule. So to minimize this, what they done is that lead compound optimized to nalidixic acid, which is 1,8-naphthoridine 4, which is naphthoridine 4. Here, this is the nalidixic acid which we told, which is naphthoridine nucleus. Now, fluoroquinolones was discovered where the fluorine atom was present at 6th position. We discussed this all things. So, why we added this? It has increased activity. Next, we can see it. Nalidixic acid. Which is having naphthoridine nucleus. What happened is that they have changed. They have brought again quinolone nucleus. Quinolone nucleus. See here the pyridine nucleus is there. That they have brought again to benzene. And they have added this, what is that? Piperazine nucleus. Piperazine nucleus, which is advantage. Because of that, see fluorine is present at 6th position, remaining all the same to same. Here there is here pyridine nucleus. Here you see benzene nucleus is present. 3 changes we have done. We have done 3 changes. So because of this, we got norfloxacin. So norfloxacin, what it does, you see, in nalidixic acid, the pyridine ring is replaced by benzene by changing naphthoridine to quinolone derivative. So because of this, what happened? There is a reduction in protein binding. There is reduction in protein binding. So there is a reduction in protein binding. There was some problem with protein binding. No, now there is reduction in protein binding. And with the advantage of what's the advantage of fluorine atom? You see, fluorine atom increased penetration to the microorganism, even in gram-negative bacteria. And it can, all these drugs, they block DNA gyrase activity. So they block this, the penetration is increased, so increased DNA gyration activity. Then introduction of piperazine nucleus at 7 increased half-life. It has increased half-life through 3 to 4 hours. 3 to 4 hours. Previously, nalidixic acid was having 90 minutes. 90 minutes, the duration of action. Now, norfloxacin, the duration of action is increased. Compared to nalidixic acid, the norfloxacin is more active. Why? These are the some of the changes. Number one is what? Naphthoridine nucleus is brought again to quinolone. The fluorine atom will help to increase the penetration, and piperazine nucleus increase the half-life. So this is how norfloxacin has been discovered. These are the advantages of various functional groups. Now, from norfloxacin, with the help of norfloxacin, we got many drugs by various changes. You see, this is norfloxacin, which we were discussing in the previous slide. Norfloxacin having piperazine nucleus, benzene, quinolone nucleus. Now, at first position, there is ethyl group. We told numbering should start at the nitrogen. So numbering, after numbering, once starting numbering, you should complete numbering to that ring, then move to the next ring. So we started numbering here. Then you can see it at the first position. What happened? They replaced ethyl group with cyclopropyl group. Remaining structure same to same. Cyclopropyl group. What it happens, you see, we got drug called as ciprofloxacin, which is 3 hours active. Even norfloxacin was also active for 3 hours. But what is advantage is that norfloxacin showed more activity towards gram-positive. Even gram-negative is present. At the same time, it also has gram-positive bacteria. Norfloxacin was having activity towards gram-negative bacteria. Now this drug has became broad spectrum. Here also broad spectrum, not much. Here also broad, but here it becomes 3 times more active towards gram-positive bacteria. By simple changing of ethyl group to cyclopropane group. Cyclopropane group. Now, one more you can see it. They synthesize many things. So here you can see it. What is that? Pillofloxacin. P-floxacin. P-floxacin. So what is the name? P-floxacin. Now here P-floxacin, what are the changes? They retain this ethyl group as such. They placed a methyl group here on the piperazine nucleus. When they place this, the duration of action, you see, 11 hours. It has increased the duration of action to 11 hours. 11 hours. Now, one more drug, you can see it. The same from P-floxacin. It was not it's not more available in the market nowadays. So what they done is that they introduced a tricyclic system now. See, these are tricyclic system. Tricyclic system. Having morpholin ring. Morpholin ring. This is morpholin ring. We studied this many times. Even in a timolol, this see nitrogen and oxygen, which are opposite to each other in a six-membered ring. It is morpholin. So based on this, we got ofloxacin. Reminding you see the same like P-floxacin. Here it is ofloxacin, where the duration of action is 6 hours and gram-positive bacteria. See, we told that these drugs are having gram-negative bacteria. Now we are increasing the activity towards gram-positivity. Towards gram-positive. So this is a now this carbon. So this carbon is chiral carbon. When it is levo in nature, what happens? We got levofloxacin. We get levofloxacin, which is 6 hours, same as ofloxacin, but the activity towards gram-positive has increased or doubled than ofloxacin. This is also one of the chiral switchers. We used to learn in medicinal chemistry chiral switcher. That means giving active molecule to the patient. Levofloxacin, levocitrizine. Instead of taking citrizine, nowadays they are marketing levocitrizine. In the same way, ofloxacin has almost replaced by levofloxacin, which is more active. The duration of action is same, but activity towards gram-positive. Already they have gram-negative activity. Gram-positive activity increased. So this is one class of drugs. Any doubt, please? Take 2 minutes. Any doubt you can ask. I'll be scrolling. Nalidixic acid converting to norfloxacin. So first what we discussed, quinoline. Quinoline is a heterocyclic ring. Quinine, generally we get confused with quinine. Quinine is anti-malarial drug. Then we have seen tautomerism. I think you people studied. But our drugs have 4 quinolones. Simply we kept here to not to get confused. 4 quinolone rings are present in our drugs. Then we have seen these drugs' importance in the clinicals. They are also very active against intracellular parasites. Nothing but atypical microorganisms. Safety is good. Therapeutic index is also good. Bactericidal, broad spectrum activity. First generation drugs are more active towards gram negative. Now, as the generation increases, they have equal activity towards gram positive bacteria. So we discussed here, it is from the chloroquine byproduct. We got 7-chloroquinolone. It was antibacterial, but duration of action and everything was less. So we got nalidixic acid. Nalidixic acid. Then various further we got ciprofloxacin. Just now we have seen these. What is the advantage of ciprofloxacin? All this we have learned, and we have classified first generation, second generation, fourth generation quinolones drugs. So these are the except enoxacin, this except enoxacin, remaining all drugs contains fluoro group on the 6th carbon atom. On the 6th carbon atom, we have fluorine group. So this is nalidixic acid. Then nalidixic acid is converted to norfloxacin. What happened is that we got good activity, gram positive activity, also gram negative also it increased. Good half-life also increased. Norfloxacin to ciprofloxacin we studied. Then even to p-floxacin, then ofloxacin. Then from ciprofloxacin, which is second generation drugs, we can see some of the third and fourth generation drugs which we got. So here ciprofloxacin, you can see it, a lot of information we have given at the same time. Now, so introduction of methyl group on piperazine. It is here now, you can see it is on the side chain, not onto the nitrogen ring. Onto the nitrogen ring. And a methoxy group. And a methoxy group at 8th position. You can see it. We have 6th fluorine group. Then which is having activity towards gram positive and anaerobes. Even bacteria which doesn't need to grow in oxygen, it also can treat, which is the drug called gatifloxacin. Now, multiple changes we have done. We can see introduction of piperazine, methyl substituted piperazine, methyl substituted here too. Sometimes you can see a substituent on the 5th, either with the NH2 group or OH. There are some drugs we didn't place, or OCH3 group is present. This all increased activity towards gram positive bacteria. Now you can see it at 8th position. 8th position, there is also fluorine atom. So it is a saprofloxacin which has fluorine atom at the 7th, at the 6th as well as 8th. Then in moxifloxacin, you can see it. The fluorine, there is no piperazine nucleus. No. We have. Wait. Numbering, you can see it. We changed differently here. Numbering starts from here. Now number 2, we reversed it. Deliberately, I reversed it so that you can learn both. Sometimes some of the textbooks write like this fashion. So 5th position. No, 3. This is 4th. This is 3. I forgot 3. This is 5th. So 6th position. Now in the 7th position, whatever we used to have piperazine nucleus, has been changed to with the help of this ring. Pyrrolidine, pyrrolidinopiperidine. Pyrrolidine, this is pyrrolidine nucleus and piperidine nucleus. Piperidine nucleus, which we got moxifloxacin. Moxifloxacin. Again, in gemifloxacin, you see here, in gemifloxacin, again, naphthoridine nucleus came, just like a nalidixic acid. Nalidixic acid, this is a gemifloxacin. You can see it. What is that? Nalidixic acid, what is the nucleus here? Or here we written naphthyridine nucleus. Yes or no, please? Even in the here also we written naphthyridine nucleus. That nucleus is present in gemifloxacin. Is present in the gemifloxacin. Have a glance on this, please. Up to here it is clear, I think. Now what we done now, there are many changes done in the same structure. So if you see saprofloxacin, there is change at 8th position as well as at 5th position. So that's why we kept one sentence like this and given some of the examples. So this is ciprofloxacin. From ciprofloxacin, we got gatifloxacin. Among the gemifloxacin, you can see the gemifloxacin, which is 4th generation. It is having naphthoridine nucleus. Again, naphthoridine nucleus came back. So what they do is that organic people, they completely synthesize. They do their publication. They submit their PhD only by synthesizing. By synthesizing. So they done that synthesis. Then when they done the activity, they found it is good, passed all the clinical. Then it came into the market. Now, gemifloxacin. Now, it. Now, completely you can make all this, whatever we studied, it's a drug design portion as well as SAR. You can see it now. What is the SAR? You can see it one by one. There is no substitution in second carbon. Any of the drug, you observe any of the drug, there is no substitution at the second position. Any substitution. Here, you see any drug, there is no substitution. Number one, there can be propane group, alkyl group also can be present, or even aryl group with difluoro can be present. We didn't see any drug, but we can get it. Then third position, carboxylic group is very important. It is for binding. So there also they didn't make any ester changes or carbamate group, anything we haven't seen any change on the carboxylic group because it helps in binding to that DNA gyrase. Carboxylic group, important. And even the keto group, did we change? This is the keto group. If you want, you can make to oximes, hydrazines, but they haven't changed. Because it is important. These two all not much changes is done on this ring. And if you give numbering A ring, B ring, if you give this as A ring because it has heterocyclic A ring, and this is a B ring, what happened? Oh, B ring, there is no much changes done on A ring. Only changes they have done at the first position. If you want, you can observe, see all this ethyl cyclopropyl group, cyclopropyl group. Here ethyl group was present in even here in this p-fluoxacin. You see ethyl group. Should be present. Now coming to fifth position. You see this is fifth. Sorry, this is fifth position. So now this is fifth, this is sixth. Coming to fifth position, you see some of the drugs has amine, hydroxyl group, and methyl group. So you can see it here. In saprofloxacin, there was amine group. In moxifloxacin, methoxy, hydroxyl group derivative, methoxy group was present. Increased activity towards gram-positive bacteria. Now, fluorine. Yes, 1 second. Fluorine increases activity towards gram-negative bacteria. Yes, anesthesia?
Can you hear me?
Any doubt?
I just wanted to say
that
on position 6 of moxifloxacin, it's not methoxy group. That's not position 6.
Yeah. It is reverse, yes. So this is a we didn't learn all the drug structures. No, some of the drugs will be having methoxy. So that is correct. What you told is 100% because 8th position this is. Then 7th position you can see it. Increase activity towards gram negative and gram positive. See, piperazine, pyrrolidine, and indolin derivative you can place. Then at 8th position, that is at this X. So whatever we are talking here, it can be nitrogen. When it is nitrogen, it is called as naphthoridine nucleus. If it is carbon, it is called as a quinolone nucleus. Quinolone nucleus. Here, after when you place carbon, you can change with fluorine, chlorine, or methoxy, or methoxy group, or difluoromethoxy group. You can change them. So these are some of the SAR. We already studied SAR here itself. So as a part of drug discovery process, we discovered each drug, how it was changes, what are the improvements, we studied. Then finally, in this slide, we discussed about SAR. So all the drug structures we didn't discuss. So don't think that we didn't discuss about any drug about this indolin nucleus. There are drugs. And also you see piperazine nucleus. If it is piperazine nucleus, it is gram negative more. Even this functional group, if it has gram positive bacteria. You can see the color. If it is in a violet, what is it? This only piperazine, not substituted piperazine. It has gram negative bacteria. Now, aerobic, anaerobic, you can see it here. If you place nitrogen, it is naphthyridine. and if it is carbon, quinolone nucleus. So this is SAR. Now, mechanism of action. Mechanism of action. These drugs block DNA gyrase. DNA gyrase, which is also called as topoisomerism. Topoisomers, topoisomerase. Isomerism, I'm telling. Topoisomerase. So, what is the function of this DNA gyrase or topoisomerase? Is that during replication process, DNA is in a coiled. It is in a super coiled DNA. It will be in the form of coiled. So what does DNA gyrase does is, it breaks this. It breaks the DNA molecule, and see, it is like this. Now it starts opening. When it starts opening, what happens? New replication can occur on one strand. Another strand, it can occur from the opposite direction. So when it is unwinding, other side of the DNA molecule causes twisting. It causes twisting. Yes or no, please? When the rope, when you try to open here, another end of the rope, it gets super coiled. Super coiled. So this opening of the DNA molecules becomes tough. Will become tough. Now this topoisomerase prevents that strain produced by this DNA gyrase. See, opening. When it tries to opening, see, it was coiled. When you try to open, somewhere at the end of the other end of the rope, what happened? It becomes twisted. It becomes twisted. Now to avoid this twisting, what happened? DNA is fragmented. DNA is fragmented. Wherever it is getting twisted, what's that? Our DNA gyrase will cut. DNA gyrase has two important functions. One is cutting, and one more important function is ligating. It will cut the DNA. So to avoid that supercoiling, then after supercoiling is removed, supercoiling is prevented, then the cut DNA is again ligated. So two functions are there for this DNA gyrase, which is also called as topoisomerase. So what is the important function? It cuts DNA and also ligates. Why it cuts the DNA? To prevent supercoiling. You can see it now. It is in this fashion. Now DNA. Somewhere it will cut. So, and make the strand as relaxed one. You can see the relaxed one. Now, when it cuts, what happens? When it cuts, new DNA molecule is synthesized. So for example, this is the strand. Now what happened? It's all coiled here. Sorry. So it is coiled like this. For example, you consider that it is coiled like this, and it is coiled. Now you consider this. It is coiled like this. Now what happens is that now our DNA will supercoil everywhere. At one portion it opens. And now what happens is that new DNA molecule is generated by DNA polymerase enzyme. And one strand is called as leading strand because new DNA is found very fast. In another strand, DNA is produced in fragments. This is called as lagging strand. Then again, this DNA gyrase ligator is there, which can mix them. Now, this is how this DNA gyrase will help in help during the replication process. One is cutting so that it prevents supercoiling. It prevents cutting, and also it ligates. After cutting, no, the DNA has to be come back again to its ligating property. Now, our drugs, they block the function of this DNA gyrase function. I told you that what are the important functions of DNA gyrase? One is cutting function, and one more is ligating function. Ligating function. Now, among these two functions, our drugs will block mainly on ligating function. It doesn't show any activity on cutting. So the cutting function is retained, but ligating function is blocked. That is the beauty of these drugs. So what it does, our drugs, when you take, it makes the DNA into fragments, pieces. So all the DNA molecules are becoming pieces into pieces. So there is no strand to generate continuous DNA molecule. Are you getting on that, please? So DNA gyrase function, I told you that. What is the important function? DNA gyrase function is that cutting and ligating. Why we want this cutting and ligating is that to prevent supercoiling during unwinding process. Even you can see it, the girls when they tie here, when they try to open at the bottom, they at the head it twists. So they try to open down and also at the even at the top. The same principle also happens here. So to prevent that, God has given this DNA gyrase enzyme. Even it happens to us. Even in our DNA molecule, DNA replication process, this DNA gyrase is present. Clear? Now, this is the what we are telling. Now, our drugs will specifically block the ligating activity, but retain cutting property. So what happens is that it cuts the DNA molecule pieces into pieces. So there is no proper strand for the generation of DNA molecule. Now, one more important thing. We are almost finishing. Now you can see it. DNA gyrase. These drugs are able to block DNA gyrase, nothing but topoisomerase 2 in gram-negative bacteria, and in topoisomerase 4 in gram-positive bacteria. So now this topoisomerase 4 is more dangerous than topoisomerase 2. Topoisomerase 4, consider that it is having two cutting properties. So the cutting property of topoisomerase is more than cutting property of topoisomerase 2. So when you block this, so ligating property is blocked, the cutting property is double. You consider that this is having two scissors at a time. So it is like two scissors at a time. Whereas topoisomerase having only one cutting property. Whereas topoisomerase 4 is two scissors are present at the same time, like a snake having two heads. A snake will be having, some of the snakes having two heads. In the same way, topoisomerase is having more cutting property because it has two cutting nature like. So it cuts very destructively. Very destructively. So that is what we told. It blocks the DNA gyrase, thereby preventing unwinding or supercoiling of DNA. Unwinding will be there. Unwinding it prevents, and supercoiling will be there. So DNA replication is stopped and causes bactericidal effect. So that is the advantage of these drugs. As we oxazolidones, we learned that these drugs block early stage of protein synthesis. The same way these drugs block early stage of replication process. So you may be having DNA gyrase, DNA polymerase, but you can't get the fragment proper. DNA cannot be formed properly. Yes, please, Axilla.
Sir, would you please repeat the way you said the activity of topoisomerase 4 is more than the one of topoisomerase 2?
Topoisomerase 2 has considered that it's a snake with having two heads. So it can inject a poisonous snake having two heads. So in the same way, topoisomerase 4 is a very dangerous what is that? Enzyme. So it can cut, suppose if this is a strand. So if, for example, you consider that topoisomerase 1, it can only cut here. But whereas topoisomerase 4, for example, topoisomerase 4, it cuts here, at the same time it cuts here. So this entire will become a separate DNA molecule. When topoisomerase 2, if you cut, what happens? This will become a straight chain like. It will be having double helix, but it is straight chain like. So here, consider this is topoisomerase. When it is cutting, no, this will become a fragment. Now, if it cuts here, it becomes this also one fragment. So fragments are more with the help of topoisomerase 4. No? Fragmentation is 4. The small DNA pieces are more with the topoisomerase 4. That's what we are telling. It's a very destructive enzyme. And I also told, don't get confused. We are only blocking the activity. I don't know how it is possible, but this is the mechanism which we got the explanation. They're blocking the activity of ligating property, but not the cutting property. So like this. Clear, madam?
Yes, thank you.
Suppose consider this is mine. So if one cut, if I do it, that is topoisomerase. Now, if you cut here and here at a time, what happens? A small piece will come. Just like our KFC piece, like it will come. So like that, it does topoisomerase 4. So this is very good drugs, very, very good drugs. So carefully we should use. That's why we can see it. Now, even after useful, we told safety and all those, some of the warnings, new warnings came for these drugs. So most of them they found after 10-20 years, you can see it. In 2008, US FDA added black box warning. So everyone who are manufacturing this, they need to place it on the label, on the box. It's a warning like this. Tendon damage. So muscles will get damaged by using this. And also in 2016, again they have given warning. So disabling and potential permanent serious side effects like involving tendons, muscles, joints. First, they're given only tendons. Now, further, they have given that. So lot of research they do it. Clinical pharmacologist, what they do, clinician gets the information from our prescriptions. So that's they are giving. Muscle, joint, nerve, central nervous system. So these are the problems. When you see the benefits, disadvantages, benefits are more important. See, when you have a lung problem, when you allow these microorganisms to grow, you will die immediately. With weak muscles, we can survive. We can survive. So still we use these drugs. So nervous system, concluding that these side effects are generally outweigh the benefits of the people. The benefits are more than the side effects. Even we discussed the same thing with the anti-lipid drugs, atorvastatin, all those. They are very good drugs. At the same time, muscle issues, kidney damage. But what to do if you don't use that atorvastatin and all those? Patient will die immediately. But if you use that muscle weakness, okay, no problem. He will stop walking. He will survive it. So like that. And again, see, in 2018, they're concerned with low blood sugar and mental health problems. We will discuss about mental health problem. There is no clear-cut any information how these drugs are causing blood sugar decrease. And maybe absorption is increased. Then we will get some doubt. Why then they're not giving us an anti-diabetic drug? So these are the some of the questions. So let us see now. Side effects, you see. Nausea, vomiting, as we know. Now CNS effects, you see, dizziness, sleep disturbances, especially it will be of CNS stimulant-like activity. We will discuss in next slide. Dizziness, sleep disturbances, confusion, seizures. Liver, you see, hepatitis, liver failure, kidney, hematuria, crystalluria are present. Muscle skeletal effect, tendons, as we discussed, weakness in all those. Cardiovascular effects, hypotension, tachycardia with QTC changes. In ECG, PQRST, those QT intervals, we observe the changes in the QT intervals. Skin, rashes, pruritus, photosensitivity. Some of the drugs are very sensitive to photosensitivity, that also we will discuss. Then glucose, disturbance in glucose. So these are the some of the side effects of these fluoroquinolones. Fluoroquinolones. Then, so this is the, what is the reason they're telling fluoroquinolones? They have sleep disturbances. That's why we told CNS toxicity. What is the reason they're telling is that, now see, this is GABA, gamma amino butyric acid. Now our drug, the carboxylic group and amino N. In these drugs, in fluoroquinolones having piperazine nucleus, has similar distance. Whatever the distance from here to here, the distance between these two, between amine group and carboxylic group in GABA is same. But what is the thing is, these are almost straight chain compounds, polar in a straight chain and polar. Now these are the bulky groups are present. That's why they're telling that these drugs will go and block GABA to bind. It's like a GABA antagonist, like actin. So that's why the patient will complain after using 1 month of 10 days like that. I am not getting sleep properly like that. So these are the reasons for fluoroquinolones acting as a CNS toxicity. And also, see, for few people, because of low penetration to brain, this toxicity is rare. But this is the reason what is they have given why we get most of the patients complaint about agitation, sleep disturbances. This is the reason they have given. Then, now one more is the there is a complex forming, just like tetracyclines. So with these drugs, when you combine with calcium, magnesium, zinc, iron, what happens? It becomes complex. It becomes complex. You can see it here. Between carboxylic group and keto group, it can form complex with the metals which are divalent in nature. Divalent in nature. So this, when it happens, no, the water solubility is not present and causes crystal urea. Crystal urea. So better not to combine with these drugs. With the calcium gluconate, ferrous sulfate. If you are suffering from anemia, if you are taking antacids, then this all will happen. We discussed about photosensitivity. Some of the fluoroquinolones, when you take these drugs and exposed to atmosphere, you get allergic reactions. That is photosensitivity. That is mainly observed with the drugs which are having a fluorine atom at the 8th position. Fluorine atom at the 8th position. For example, lomifloxacin. Among all these, the ones which have halogens at 8th position will produce this phototoxicity. Among all these, lomifloxacin is considered most phototoxic fluoroquinolones. The ones which doesn't have, among them, moxifloxacin. Moxifloxacin is the least producing photosensitivity. So between, if there is functional group here, methoxy group is good. So moxifloxacin is the least photosensitive drug. So these are the some of the items which you need to remember. Then ciprofloxacin, you can see it is available in tablets, injection form it's available, even it is available in ophthalmic preparation. Very, very good drug, wide range of activity. You can see diarrhea, typhoid. Most of these drugs, ofloxacin, ofloxacins are very good against typhoid drugs. Nowadays, even they are using in TB. When other drugs are not used, not able to treat, we are using these drugs. Uncomplicated and complicated urinary tract infections, urinary infections, cystic fiber, conjunctivitis, it is given. And so these are the things, ciprofloxacin. Then lomifloxacin, levofloxacin. This is also very good drug. Levofloxacin is widely used. You can see it's a broad spectrum fluoroquinolone, and here you can see piperazine nucleus. It has both gram-negative, gram-positive bacteria. Now uses, you can see for prostatitis infection. During prostate gland hyperplasia, what happened? Urine starts accumulating, so you get infections. So in that condition, urinary tract infection, cellulitis, anthrax, endocarditis, meningitis, traveler's diarrhea, tuberculosis, plague. It's a wide range of uses are present on this. Levofloxacin is levorotatory, you know that. So these are the uses. Then one more, This is what we were discussing. Inoxacin. So listen here. This is inoxacin, which is having naphthoridine nucleus. Naphthoridine nucleus. So one more drugs are there, that is sinoxacin, which is having quinolones. Here both nitrogens are present on the one ring. Quinolones, naphthyridine, and quinolones all are bioisosteres. All are bioisosteres. Nowadays, we are not using these drugs. Maybe in some examination, they may ask. So I kept here. They may ask this. So they may ask this. So we kept. Bioisosteres means what? Drugs, functional groups which are in similar nature will also produce similar biological activity. So those are called as bioisosteres. So 3 rings are bioisosteres. Quinolone, 4 quinolone, all these are quinolones. Quinolones, we are discussing about 4 quinolones, not about second or anything. 4 quinolones. And naphthyridine and quinolones. Quinolones. These are the drugs. Then you can see its activity. So agar diffusion method, you can do it. Broth method, minimum inhibitory action, you can check it. That is what we were doing in our lab. Even with the UV spectrophotometer, you can do it at lambda max of 278 nanometer. Then even with IR, these are the some of the peaks with IR where you can observe. So this is any doubt, please? Okay, then if you don't have any doubts, we can close the meeting. Bye.
Bye, sir.
We have lab. Please come to lab.
Prof, I have a question.
Yeah, please.
It seems like most of these quinolones, they're actually inspired by alkaloids. So as much as I know you said that it comes from the inspiration of chloroquine, but it
Yeah.
looks to me like most of them are actually alkaloids.
The quinoline nucleus is present, not from quinine. No, it is chloroquine.
Okay.
Chloroquine is a synthetic anti-malarial drug, not a quinine drug.
Okay. All right.
Maybe when we discuss regarding anti-malarial drugs, we will tell how from quinoline chloroquine came. Just a splicing of upper ring we will discuss there.
Okay. All right. Thank you.
Yeah. Any doubt, please? Sign for your attendance. All are present, no problem. 33 was there. I can no problem. I will give attendance. If you don't sign also. Bye. I'm closing.